Interphase. DNA firstly starts off as freely floating chromatin in the nucleus before condensing into chromosomes. Currently, there are 6 chromosomes in the model, and in humans there would be 23 chromosomes. In the S phase of interphase, DNA replicates to prepare for separation. These new copied chromosomes are attached at their centromeres, forming sister chromatids. The centriole, the spindle fibers that pull chromosomes apart during anaphase, also doubles in preparation for division. At the end of interphase, there are 6 chromosomes in the model, and in humans there are 23. Prophase 1. Tetrads, or pairs of homologous chromosomes each with 2 sister chromatids, are formed during synapsis in which they fuse together. There are 4 total chromatids and 1 tetrad. Chromatids of the tetrad line up and cross over with each other at chiasmatas, or locations of contact. Crossing over shares genes from one chromatid between the other, a process that happens 2 to 3 times for humans. Centrioles move to opposite poles of the cell, where spindle fibers begin to form. Lastly, the chromosomes begin to move to the center of the cell, otherwise known as the metaphase plate. Metaphase 1. Spindle fibers begin to connect with the centromeres of each homologous pair, which are arranged on the metaphase plate at random. There are 8 different combinations of assorted chromosomes in the model, and 8.3 million different combinations in humans. Independent assortment is genetically important because it creates random formations of DNA, which creates genetic variation that is important for natural selection. Anaphase 1. The spindle then disconnects the centromeres of the homologous chromosomes and pulls the sister chromatids, which are still attached to each other by their centromeres, towards the opposite poles of the cell. This contrasts with mitosis because anaphase in mitosis separates sister chromatids, while anaphase 1 in meiosis separates homologous chromosomes. Telophase 1. The spindle fibers continue separating the chromosomes until they reach the poles of their respective spindle. 2 daughter cells then separate and form. At the end of meiosis 1, there are 6 chromosomes in the model and 23 in a human body in each cell. This is the same as the starting number of chromosomes because the cells separate at the end, which causes the doubled amount of DNA to have. Cells after meiosis 1 are haploid cells because they have half the amount of DNA as their parent cell. Prophase 2. A new spindle forms in the daughter cells and the sister chromatids move towards the metaphase plate. Metaphase 2. Sister chromatids finish lining up with each other in a straight line on the metaphase plate. Anaphase 2. The sister chromatid centromeres are disconnected and the sister chromatid is separated, then pulled to the opposite poles of the cell. Telophase 2. Nuclei form at the spindle fibers of the cell and the cells separate. The amount of chromosomes in each cell is 3 chromosomes. This is half the number of chromosomes at the start of meiosis 2, because there is no DNA replication in meiosis 2. The cells resulting from meiosis 2 are haploids, because they have half the DNA from their parent cell. The primary sources of genetic variation are from crossing over, independent assortment, and random fertilization. This is important because genes that better fit an environment will mutate and be passed down, whilst other unfit genes will die out. It is important for natural selection, so that genes can mutate to adapt to an environment.